Okay, so now we're going to discuss how glycogen synthesis and degradation is regulated, which is, of course, very important for both maintaining blood glucose levels as well as for work, for functioning of your muscles. So, the regulation of glycogen is going to be dependent on what the tissue needs. Liver is going to use glycogen during the fasting stage and between meals to support blood glucose, so it's going to help to maintain blood glucose levels. Regulation, therefore, in the liver is going to be on insulin and glucagon. Insulin, of course, will always override glucagon, but during the fasting state and between meals, glucagon is really going to drive glycogen degradation. Now, degradation in your liver is also going to be activated by epinephrine in the absence of insulin as well, so as needed, fight or flight type of situation. So epinephrine can also stimulate the breakdown of glycogen stores. Now, in the muscle cells, your glycogen is going to be used for ATP for the actual muscle cell. So it's going to be regulated mainly by need. So the presence of increased AMP. AMP is an allosteric activator of glycogen degradation, as well as during calcium. So calcium is going to be released when you do exercise, when you do work. And so during muscle contraction, calcium will trigger the glycogen to be broken down as well for a source of energy. As with your liver, epinephrine can also trigger your mobilization of stored glucose in the form of glycogen and breaking down those stores. One thing to note is that the actual glycogen stores in the muscle during fasting will decrease very little. So we'll see this again as we go through the regulation that the muscle is not sensitive to glycogen. It does not, it's not sensitive to glucagon, does not have glucagon receptors, and therefore glucagon can never stimulate the breakdown of glycogen stores in a muscle cell. So here's just an overview of glycogen regulation. So again, it depends on where we are. So the liver versus muscle. And so the liver, of course, is going to maintain blood glucose levels. So during fasting, you're going to see your degradation of glycogen is going to be activated. And your glycogen synthesis is going to be reduced, of course. And this is just so that your liver can release free glucose so that your red blood cells, your brain, can use those glucose stores as for energy. After the carbohydrate meal in the fed state, of course, it's going to be reverse. This time your glycogen synthesis is turned on, your glycogen degradation is turned off. Insulin is going to be driving this primarily. In the case of glycogen degradation, it's primarily going to be glucagon. And then when you have exercise or stress, epinephrine is going to cause the degradation of glycogen and a decrease of glycogen synthesis. Also, calcium calmodulin can have the same effect. Now, when we go to the muscle cell, as you see, we don't have any glucagon signaling, but as your blood insulin levels drop, you see that you have a decrease in glycogen synthesis, but there is no degradation of your actual glycogen. During a fed state, when insulin rises, you have your synthesis increasing, and during exercise, when you have epinephrine or calcium calmodulin or AMP, then you're going to have glycogen degradation is going to be turned on. Now, in the case of the liver, what's really important is the amount of fasting. So the breakdown of glycogen stores is going to be pretty constant for almost up to 24 hours, almost a full day. When you hit that 22 to 24 hour period, you see the rate of degradation, of breakdown of glycogen drops dramatically. So up until this point, it's going to stay relatively constant. And it's just because you have only so many enzymes that are working on the non-reducing ends, and they're breaking down glycogen until you start to run out of glycogen. And then your rate of glycogen genolysis is going to decrease. Now, in the liver, you're going to need to maintain blood glucose levels, so that's the main purpose of your glycogen stores. The liver does not use glycogen for energy. And so your insulin-glucagon ratio is going to be key for your liver cells. Now, if you have an increase of blood glucose and you don't need to release new glucose, your glycogen degradation is going to be inhibited and you're going to stimulate the building up of glycogen stores. Now, when you have a fall of the insulin-glucagon ratio, this is going to activate degradation of glycogen to release free glucose into the blood. One thing to realize about, again, we're in the liver, is that we're going to break down glycogen stores as well as make new glucose via gluconeogenesis. These are going to be activated together at the same time. They're both going to help to maintain blood glucose level during the fasting period between meals. However, glycogenolysis is more rapid because you already have glucose made. All you need to do is take it off of the glycogen molecule, release the free glucose, whereas with gluconeogenesis, you actually need to make a whole new glucose molecule. So let's talk a little bit more about the regulation. So we're going to have your glycogen phosphorylase B, which is inactive, and glycogen phosphorylase A, which has a phosphate, is going to be activated. Now on the other side, we're going to have your glycogen synthase independent, or A, which is active. You don't need to be modified. And then glycogen synthase D, or dependent, or B, a little B here, is inactive, and this is phosphorylated. So it's important to note that the breakdown of glycogen, so here we have glycogen breakdown, and phosphorylation activates this enzyme to break it down. And then we have synthesis of glycogen, and dephosphorylation activates the synthesis to build it up. So they're kind of alternatively phosphorylated, the different pathways. Phosphorylation will turn on the breaking down of glycogen. Dephosphorylation via phosphatase will induce the building up of glycogen stores. Now, regulation is going to be critical. Again, we're going to have in the liver, phosphorylation state of enzymes is going to be critical. We're going to have your glycogen synthase, in which you're going to have a phosphate that inhibits. You're going to have your phosphorylase, phosphate activates, just as we just said. We have phosphatases that are important for removing phosphates, and in your liver you have hepatic protein phosphatase 1. So let's just blow this up just a little bit. And so in here we have, of course, depending on fed or fasting state. So in the fed state you will bring in glucose, and your glucose will be converted to glucose 6-phosphate. Your glucose 6-phosphate will become glucose 1-phosphate to UDP glucose, and then you're going to build up glycogen stores. So in this case, your glycogen synthase is activated, and over on the other side, your glycogen phosphorylase is going to be, where is it? Right here, inactivated. Now there are a few steps in between all of these. So right here we have a glycogen phosphorylase kinase. And so when it is dephosphorylated in the presence of insulin, the protein phosphatase, it's inactivated, so you don't break down glycogen stores. In the presence of glucose and protein phosphatase, you're going to activate your synthase and you're going to build up glycogen stores. Now the opposite is going to happen, of course, when you have your glucagon or epinephrine signaling. And in this case, you're going to get cyclic AMP, and this is going to activate protein kinase A. And then protein kinase A is going to phosphorylate phosphorylase kinase. Phosphorylase kinase is now active, and phosphorylase kinase is going to phosphorylate glycogen phosphorylase. And this is now going to be active. And then you're going to start to break down glycogen stores. Now also what's happening is your protein kinase A is going to phosphorylate the synthase and make it inactive. So you're not going to build up glycogen stores. So they're going to be alternatively activated. Now, regulation, of course, is going to be very critical. So in the liver, it's important to note that insulin is the major regulatory hormone and reverses all the effects of glucagon as well as the effects of epinephrine. And so insulin is the most important hormonal regulator of blood glucose. You have greater changes of insulin levels during fasting than you do have in glucagon. and therefore insulin is regulator of glycogen synthesis and degradation. So the presence of insulin will promote synthesis, the absence of insulin will promote degradation. Insulin is going to regulate hepatic protein phosphatase 1. This is also phosphorylase A phosphatase. This is through that tyrosine kinase receptor cascade. Insulin also activates the phosphodiesterase. This is going to convert cyclic AMP to AMP, and this leads to inactivation of pKa. Protein phosphatases are going to bind proteins that target phosphatase to the glycogen particles, and there are 4 different proteins that are targeted by protein phosphatases. You have G, which stands for glycogen, M in the heart and skeletal muscle, GL in the liver. You have regulatory 6 proteins in most cells and regulatory 5, as well as protein targeting to glycogen, PTG. And protein phosphatases will target these proteins, and these proteins bind to the same region on protein phosphatase. So one protein phosphatase can only target one of these proteins. Now, during the fasting stage, protein phosphatase 1 is inactivated. This is by dissociation from the actual glycogen particles, so the substrates, the enzymes, are not available to the phosphatases. There are also inhibitor proteins, such as inhibitor 1, and this is going to be phosphorylated by glucagon or epinephrine that can then in turn regulate protein phosphatase. Now, epinephrine and calcium is important in our muscle cells, but it also has a role in our liver. So epinephrine or adrenaline is the fight or flight hormone. This is going to be released by your adrenal medulla, and this is going to promote the increase in demand for glucose for mobilizing fuels. So epinephrine will stimulate glycogenolysis in the liver by the alpha and beta agonist receptors. Your beta agonist receptors are going to go through that same story we've already talked about: G protein, adenylate cyclase, cyclic AMP, protein kinase A, etc. And then alpha receptors is an increase of calcium in the liver via phosphatidyl and Oxatol bisphosphate signaling transduction system. So here's the phosphatidyl and Oxatol pathway. Again, it's always going to start with epinephrine, and in this case, we're having an alpha receptor. It's still associated with a G protein. It's a G protein receptor, G alpha. We then are going to get activation of phospholipase C. Phospholipase C is then going to convert phosphatidyl and Oxatol to diacylglycerol, which stays embedded in the membrane, and you release IP3. IP3 will go to the endoplasmic reticulum and will stimulate the release of calcium. The release of calcium activates protein kinase C, and protein kinase C is what's going to cause phosphorylation and inactivation of your synthase. Calcium will also bind to calmodulin, and calcium calmodulin will activate the calmodulin-dependent protein kinase, which also can phosphorylate and activate the synthase. Calcium calmodulin will also activate phosphorylase kinase, and this will phosphorylate and activate glycogen phosphorylase A. So then you start breaking down glycogen stores. Now, glycogen synthase is going to be regulated by glycogen synthase kinase 3. Glycogen synthase kinase 3 is going to inhibit glycogen synthase via phosphorylation. So we have here glycogen synthase kinase 3, and we have our protein phosphatase 1, which is activated by insulin. So insulin will block glycogen synthase kinase 3, and insulin will activate protein phosphatase 1. So glycogen synthase is inactivated when it has phosphates added to it. The phosphatase here will release that, will dephosphorylate and activate your synthase. Your glycogen synthase kinase 3 will add phosphates and inactivate your glycogen synthase. So glycogen synthase kinase 3 is the kinase that will work opposite of your protein phosphatase 1. So a little bit more about glycogen synthase kinase 3. So glycogen synthase kinase 3 inhibits glycogen synthesis. It adds a phosphate to that synthase and inactivates it. There are two homoenzymes, GSK3A and 3B. There are over 40 proteins that are going to be phosphorylated by glycogen synthase kinase 3. When glycogen synthase kinase 3 is phosphorylated, its activity is inhibited. So it is going to be regulated by phosphorylation itself. Glycogen synthase kinase 3 can be phosphorylated by all of those downstream kinases that are activated following insulin signaling. Now, there are other pathways that are important for this, and there's something called Wnt signaling. And Wnt signaling has an association with colon cancer. We have patch smoothen pathways. These are regulatory pathways that, when they get disrupted, are also going to be associated with cancer. And then there's also microtubule phosphorylation during the process of apoptosis that is going to be dependent on glycogen synthase kinase 3. Now, insulin is going to phosphorylate this directly via that AKT, and then loss of this activity is being actively researched as part of a role in type 2 diabetes as well.